All right, thank you very much for the invitation. So what I'm going to do is tell you about work in my laboratory. It's really to take various kinds of high-throughput data and obtain mechanistic models of what's going on in the cell like this. I work largely with three people: Huang, who's currently a graduate student in the lab, and who were postdocs in the lab when they did this work. The overview of the talk, first I'm going to try to convince you that there's a problem in how we think about omic data, and tell you about these two algorithms that we published last year for analyzing omic data to try to achieve what we talked about in that early slide, to get more mechanistic understandings of what's going on. And then I hope to have time to tell you about some what I think is very exciting progress, taking these algorithms which we show useful in yeast and showing that we can actually do quite a lot with them in mammalian systems. So when I think about omic data, I often think about this classical reference, the Oracle at Delphi. So the Oracle at Delphi was famous in the ancient Greek world because people would travel great expense and over long distances in time to get there and pose their questions to the priestess at the Oracle. And she would give them an answer, which in hindsight would always turn out to be incredibly accurate, but at the time they rarely understood what it meant. And I think this is a good metaphor for omic data, where we spend a lot of money time collecting it and we get this list of genes when we don't. For those of you who like lower brow cultural references, you might think of Hitchhiker's Guide to the Galaxy, where they build this computer that takes 7.5 million years to compute the answer to the ultimate question of life, the universe, and everything, and they get an answer, and what is it? It's 42, and then they realize they didn't understand what the question meant. So when we do an omic experiment, we take some cell in a condition of interest. Maybe it's a gene that's been activated or repressed in some cell, or maybe it's a some found, and maybe I do a microarray and you do a mass experiment, and she does a genetic screen, and we can only guarantee that our answers are not going to overlap very much. And the data disappear. These data are examples of a phosphoproteomic experiment on the same cell. These are mammalian cells, and RNA is completely correlated with change in phosphorylation. The right shows many different genetic genes when either overexpressed, modulate the phenotype of a cell, I should say, are naturally overexpressed in those conditions. And those do overlap, not statistically significant. It is that each of these assays is asking a different question. What is the question a gene's answer? As I said, overexpress a particular gene. How do you look at expression data? So, different in yeast was that enriched transcription-related signal transduction molecules, and these represent genes that are different expressed. Doesn't mean the things that are enriched in expression data tend to be metabolic processes, and in turn poorly represented in the genetic data. So this gives an idea that perhaps if we took genetic data and expression data in the same condition, we could actually construct a more comprehensive view of what's going on in the cell by looking for interactions in which the genetic hits were actually upstream of the expression changes. And in the process, then, we could not only understand the mechanism by which these genetic hits influence gene expression, but we could actually identify some of the components shown in red in this hypothetical signal transduction pathway that were not detected in either screen, but clearly are the missing pieces of the puzzle. So to do that, we need some way of connecting these expression data back to the proteomic level. And we, fortunately, in yeast, have plenty of chip-chip and sequence analysis techniques that allow us to map expression changes back to the transcription factors that cause those expression changes. And then we can use protein-protein interaction data to link that up to the rest of the signal transduction pathway. The data that we use then that are not condition-specific are in an interactome, which come from high-throughput studies, and some of which plus protein-DNA interaction that we published, where we've actually experimentally analyzed almost every genome. So with those data, we move to an array that we treat we get from microarrays as distinct entities from the proteins that they encode. All right, this shouldn't be controversial, but other people do it other ways. So in our models, the transcripts are only connected to the proteins that drive the transcriptional change, transcription factors, and not to the proteins that interact with their protein partners. Now here's a simple test case where we have a well-characterized pathway for which there's a perturbation, for which there are published genetic data and different expression. And we can ask whether, in fact, as we expect, these genetic hits lie upstream of the differentially expressed genes. And whether the network that we can construct using those protein and protein DNA interactions really recapitulates this. So here's good news and bad news. The good news is that you can certainly connect these in the interactome, but unfortunately what you get is one of these hairball diagrams in which you've really gained no useful relationship between the genetic data and the expression data. So what are we doing wrong? Well, it's pretty an interactome like this. You've got both high quality interactions and things that are more in nature. And so we need to treat our interactome probabilistically. And we also need to be aware of the fact that while some of these genetic hits are going to lie upstream of transcription changes, some of them are going to be involved in processes that are not represented by a transcriptional output. Some of them are going to be false positives. So we need an algorithm that allows us to not connect all the genetic hits to all the regulated genes, but just some of these to some of these. And of course, it would be nice if it put all sorts of constraints on how big the network can be, et cetera. So the approach that we took to generate a probabilistic interactome is not original. We followed the path of these more groundbreaking papers, which showed very clear ways of using Bayesian statistics to assign a probability to each edge based on the reliability of the underlying data. And so we can imagine a hypothetical example like this, where we've got a genetic hit, and through the interactome, it can be connected to these three differentially expressed genes. Some of the interactions are high quality, one is low quality, and we want an algorithm that can allow us to choose which of these is connected. And so intuitively, we'd expect that this short high probability path will be more probable than this path that has a low probability edge. The approach that we took to doing that is to use a minimum cost, maximum flow algorithm, which I'll explain now. One way to think about it is to think of each of these edges as representing if you've got a high probability edge, then it's a thick pipe, and if you've got a low probability edge, it's a thin pipe. And so we connect this genetic hit to a source of some imaginary flow. As it flows through the network, it's going to go more through the high quality edges and less the edges. And this then is a ranking that we can use to decide which of these, differentially genes is likely to be connected to this one, and which of them we're not detecting as relevant to our process. So for each edge, the probability will convert that probability, taking minus probability, so lower probability means higher flow going through. To minimize this cost, well, so we max the flow. And when you do this, then networks. So instead of getting results like this, you get this result, which connects many of the genetic hits, but not all of them, to many of them. Result hit nodes that make a lot of sense, be regulators of the main response pathway and rich for the expected pheromone response annotations. But our goal is not simply to learn about you, about here some of you may have heard her on Sunday, and she's looking at how this somewhat mysterious protein that's been identified in the Parkinson's causes cell death. So this shows up in the brains of these patients, but it's not really known what it does. When you express it, it's to kill off the yeast cells in a way that's reminiscent of these diseases. There's a dosage-dependent effect and very action reactions issues and so on. So you can apply all the tools which you heard about in the previous talk in a screen, and the screen to find genes that when overexpressed the toxicity of the some her screen showed enrichment cases and effects. We need to do this now expect based on what I told you previously on completely different results, which is about what you completely different, so we can apply what we call response net, but if we apply response net, we go network that leads us to a number of one I think illustrates different richness in the this piece of the network that regulates many of the different proteins. Most of these mitochondrial proteins, so the expression data are enriched for changes in expression of mitochondrial proteins because it emulates all of these. Nerve test predictions of what alpha-synuclein is doing. Modulators act that we did a number of tests, and you could tell of biological validations. That algorithm is very precisely designed to try to understand what the connection is between different extremes. We're really finding how one data set the connections between this and this. Not optimal. Imagine, for example, a lot of proteomic data from you try to force connect between early proteomic events and changes. You have to go through a lot of different connections, and you'll more like something useful out of them. So different both connections between among data sets. Similar relation that each edge is associated with a cost, and that proportional to the probability. But now we actually pay a penalty if we keep a node out of the solution, and the amount that we pay out of the solution is how confident we are it showed up in the experimental data. Many of these experimental hits, whether they be phosphoproteomics, change each other through the network. And the algorithm is not included. It's a penalty. If we do include it, it pays a cost. That allows us to decide, or I should say the algorithm to decide, whether the sire like this is worth being affected. Is it worth paying the edge costs or T for exclusion? We applied this where it was phosphoproteomic data, 100 close to mating pheromone. So it's clear there's a lot. Interestingly, more than half of you naively try to change expression and gives you very response pathway. Ones that are hidden nodes but are clearly part of the pathways, which in hindsight proteomic changes cycle as cell structure in response pathways, but instead of the we designed this with a grants people serve humans. And it is in interaction. Cell shadow we have in yeast cells where we think almost reach transcription factor. But what I'll show you right now is that we can actually overcome these problems. I'll show you largely how we've overcome the problem, but in fact also have very interesting results where we've been able to overcome the limits of our novel transcription relation as well. So the problem we're working on is the mutation receptor, and it's a glioblastoma. That's a brain tumor that killed Ted Kennedy. It's one effective treatment. Higher levels of this mutant receptor prognosis. We had a collaborative phosphoproteomic on sequencing and that control try and understand those proteomic changes since from the string base. This color in them are differentially phosphorylated. There are a number of subdecays that these evidence that glutamate receptors. What I think is really interesting strategies. You can look at which nodes are most central to the network. You might imagine if these are actually real represent or related to the reality of signal system, that if you were to hit the most central nodes in these networks, then you would have the biggest effect on the signaling, and therefore you might be able to influence treatment. So how do you determine a centrality? Well, remember that our edge that it doesn't need to actually solve the problem of those edges. We're looking for the nodes shown here, a circle. You can see that's dark here. Well characterized and involved. Before we cancer research working with, and you oncogenic simultaneously inhibit E kinases, you get much more dramatic, so that when these cells are placed, you actually extend the lifespan inhibiting this node and the EGF receptor. So the most central node, the second on the I'll just show you that in addition, grab2 is part of targeted because large against that as being a mechanism. YW showed me that, and the fifth most central node promotes migration. So technologies that we showed were relative to proteomic and genetic systems, and strategies. People involved, I think I've already flow algorithm that Carol developed right and Synquist.
So I'm going to ask, and I know that they get more action, so on and so forth.
So I think that you get back are the we actually add back. It's a really good representation. I believe that of perturbations.
These studies is how do you
There are
so many hubs work essentially because
don't correct for that. The central nodes here expected that we would just get. I think it's because the phosphoproteome. Those data actually can look at hubs that aren't connected. Answer.
Of the protein of intervention are basically cancer targets. So
And we're at risk of those part of the network to look at pieces of the network, but it's interactions. Protein. We're always going to downweight the experimental result.
You have considered for data.
Yes. I think it should be applicable.
Data set, ISMB.
And I think the advantage. Previous talk, you said, the right, and so I think this is a lot of weak signals. Individual hit.
Are you just knowledge engineer and develop approach?
I'd be very use linear programming. The airline industry uses them. Our code to people who and figuring out how to do the interaction to try to help figure out how. I understand that there are some to slot